Hey everyone, Mr. Murphy here. We're going to wrap up Chapter 5 with the final section all about signal transduction. Signal transduction cascades, that's the main point of this section, and it's kind of the culmination of this entire chapter. So the goal here is to understand now what happens inside of the cell once a signal has been received, and when that signal then leads to all the other things happening inside of the cell. There's some important details to take away from how that happens to understand how it can achieve major cell response to relatively simple stimuli. So in this process, there's a number of secondary messengers that are involved, and these are basically intermediates from the receptor to the whole cascade of responses. And the example that we're going to use is epinephrine, which is adrenaline. And it's part of the fight or flight response. So basically, like when a shocking thing happens, like there's a sudden loud bolt of lightning, or somebody startles you and you feel that adrenaline rush and your whole body kind of feels uneasy. What's happening is your body is responding to a potential threat and preparing you to deal with it. That's the idea of flight or fight. The idea that you're either getting ready to run out of there or you're getting ready to fight off some sort of bear that's broken into your home. The process, again, pretty simple with a little epinephrine being received by your liver cells, but it's going to lead to the massive breakdown of glycogen into glucose, pouring that glucose into your bloodstream and allowing you now the energy you need to prepare for whatever event has occurred. And just think about any time you've been startled, how quickly you go from like the sudden shock to like that uneasy feeling. Just gives you a sense of how quickly this whole chemical cascade can occur. So it's a long process, and this chapter details sort of the step-by-step way in which researchers have come to understand this, but basically summed up, it's the idea that by testing different cells and exposing certain protein components to others, we can piece together the functionality of these individual proteins, and now we have a really clear picture of how this works, at least in this specific example. There are countless signal cascades that the precise mechanism is not yet understood. But what's kind of cool is, like we've been saying, as long as you understand sort of the big picture of what's going on, we can make some reasonable assumptions about how things probably happen, which will later on be inevitably proven to be pretty spot on, because as long as we understand the broad cause and effect and we have a good grasp on how chemistry mediates these things, we can make predictions about what those intermediate steps are all about, even if we don't precisely understand exactly what protein is allowing it to happen. So the molecule that's sort of relevant to this epinephrine example is the second messenger called cyclic AMP. And that AMP is adenosine monophosphate, like as compared to diphosphate ADP or triphosphate ATP. And these messengers allow the cell to actually amplify the response by doing their jobs multiple times, and we'll see that in a moment. But here you can see adenocyclase, which is an effector protein. It's a embedded protein here that would be activated by a G protein linked, or rather by a G protein, and would lead to ATP being broken down into cyclic AMP and a couple of phosphates. So this all involves many steps, and we're going to walk through the liver cell example of epinephrine stimulating a G protein-mediated protein kinase pathway, which, what a mouthful that is. But basically what we're saying is that epinephrine binds to its receptor, activates a G protein, cyclic AMP gets produced, and then activates a whole bunch of other things, which can lead to both inhibition and activation. Excuse me. So, here it is. Epinephrine is a ligand. It binds to a receptor. The receptor changes its shape, activates a G protein, because it's a G protein linked receptor. The G protein is activated and causes the activation of adenocyclase. This produces cyclic AMP. Cyclic AMP causes the activation of protein kinase A. Protein kinase A does two things. One, it shuts down glycogen synthase, and we'll come back to why that's important, but it also activates phosphorylase kinase. And now would be a good time just to tell you, don't be overwhelmed. I will not ask you about the names of any of these intermediates. I might show you this diagram and ask you questions about it, but It's just not a valuable thing to be memorizing every little intermediate protein when the big picture is all about how this process can happen in a million different ways depending upon what the body's responding to. But the point is that it works. This series of this protein becomes active, and now that it has a new shape, it can then change the shape of this other protein, and so on and so forth. This continues then. Phosphorylase kinase activates glycogen phosphorylase. Glycogen phosphorylase now does the actual job, which is taking glycogen and releasing a glucose. Even that's not super simple. It's a more step-by-step process. But a glucose is liberated, delivered to the blood, and now you have one more glucose, right? We're going to reverse, and we'll see how it's a little more powerful than that. First off, when the cyclic AMP activates protein kinase, I mentioned that protein kinase shuts down glycogen synthase. That inhibition step should make sense if you think about the name of glycogen synthase. It's synthesizing glycogen. Well, the way in which that would be happening, think back to like chapter 2, glycogen's a whole bunch of glucoses strung together in crazy branches. So glycogen synthase is taking glucoses and storing them. Well, now we have this event where we want to be releasing glucose. So one of the steps in this, in addition to just releasing glucose, is halting the process of capturing that glucose, because you don't want the body fighting itself as it's trying to release glucose and simultaneously trying to capture glucose. That kind of would negate. So we shut down the glycogen synthase. In addition, pay attention to these numbers on the left-hand side. This is the amplification happening. A single epinephrine molecule can lead to the activation of this G protein, which activates adenocyclase. That can lead to 20 different cyclic AMPs being created. So it's not just it breaks down one ATP, it breaks down 20 ATP on average. That cyclic AMP, or each of those cyclic AMPs, can lead to protein kinases being activated, and each of those protein kinase A's can lead to 5 additional phosphorylase kinases before the protein kinase itself is no longer active. So if we had 20 cyclic AMPs and 20 protein kinases, each can make 5 of the next thing. Now we have 100 of these phosphorylase kinases. Well, each of those 100 phosphorylase kinases can activate 10 of these glycogen phosphorylases. So now we have 1,000 active glycogen phosphorylase enzymes inside of the cell. Each of those can liberate 10 glucoses on average before they're deactivated, which means that those 1,000 active glycogen phosphorylases can lead to 10,000 glucose molecules. So one epinephrine leading through this cascade where each individual step, or not each individual step, but many of these steps can do their job multiple times before they are inactivated. One epinephrine can lead to 10,000 glucose molecules being dumped out of the cell. And so this is how when you get that sudden rush of energy, it's so quick because every individual epinephrine molecule that is released and finds its way to a liver cell is able to stimulate the release of 10,000 glucoses, achieving a very quick response and a very dramatic response. And that's why you feel so uneasy after you've been startled, is because your body just took like as much glucose as it could and jammed it into your bloodstream. And, more often than not in our nice, comfy human lives, we get startled and it's like not something you actually need to respond to. And so then you're just sitting there and your blood is just like full of glucose, and that's why you feel really uneasy until this process no longer is producing epinephrine, because the startle reflex or response is no longer happening, and now your glycogen synthase is allowed to activate again because it's no longer inhibited, and then the glycogen synthase is just recollecting all of that glucose and producing the glycogen again, and that's how you calm down ultimately. So that process of inhibition is important because you want to prevent the normal thing from happening while simultaneously activating this alternative pathway. And this is common in signal transduction, which is to say common in events where the body needs to respond to some sort of like notable change in stimuli in order to achieve like a new action from the cells compared to the normal homeostasis-seeking stuff that the cell is just trying to maintain the status quo. And that's why, we as humans can do like more interesting things than just like sit and exist. We can respond to stimuli by activating and deactivating different pathways in order to allow ourselves to accommodate the fact that we get different stimuli and we want to do different things, even though really at the end of the day, not that cells actually want things, but if they did, all they want is homeostasis. They just want to sit there and do nothing and exist. And I know that sometimes when you're listening to a whole bunch of AP Bio YouTube videos, that's probably what you want to. And I feel for you. I do. The response shuts down once everything has been basically reverted back to normal, which should make sense, but also carries with it the implication that there have to be proteins that are responsible for doing that reverting, right? There has to be separate proteins now that are taking all of these activated things and restoring them back to their inactivated state. And here's an example of two of these. So like a protein kinase that's been activated is going to be active until another protein comes along, protein phosphatase, and rips off that phosphate group and says, no, go back to being inactive. Just like with G proteins, they become activated when that GTP is there until GTPase comes and says, no, go back to being GDP and stop doing your thing. This is where, again, an idea introduced in an earlier video, the body deals in concentrations. And so when we are responding to events, there's a concentration shift where the activators suddenly outnumber the deactivators. And then when the stimuli goes away and the activator level kind of comes down, the deactivator level by comparison is higher and reverts things over time back to their resting state. And so that's another example of homeostasis. The body it's seemingly fighting itself, but really it's just mechanisms that allow for a balance to be struck so that we don't go like all in on just like do the thing. And then at the end of the thing, like it's, this impossible task to revert things. It's a balancing act between synthesizing the response or shutting it down. So this can happen in different ways. We can control the synthesis or breakdown of the enzyme, or we can activate or inhibit the enzymes through other molecules. So there's a variety of ways in which all of this can be controlled, but the point is that at the end of the day, it is going to be controlled. Cell functions change in response to environmental signals all the time, not just hormones, but also when ion channels open, sodium, potassium channels, chlorine channels, calcium channels. All of these things can lead to a response inside of the cell, which can drastically alter the functions of different proteins inside of the cell. Alterations in gene expression, so controlling transcription, translation, and how we are ultimately producing proteins inside of the cell can be controlled through mechanisms like this. And then also just changing the enzymes activities as well by producing inhibitors or activators or cofactors involved in the enzyme function. So all of these things can be controlled through these different cascade events. At the beginning of this chapter, it presented a question about how does caffeine basically lead to its effect, and had mentioned in the first video that it's a pretty good encapsulating question of like all the ideas of this. So caffeine is a polar molecule which can stimulate response inside of the cell through a signal transduction cascade, or actually by inhibiting what would be a signal cascade. So it turns out that caffeine structure is very similar to adenosine. And adenosine is a molecule, a signaling molecule that basically shuts down activity or prepares the body for like a restful state. It's what makes you feel drowsy. And what caffeine does is it just gets in that receptor and inhibits the activity from actually occurring. And it does this because it has a very similar shape to adenosine, and so it can sort of fit into this receptor, but it's different enough in its shape that it doesn't cause the correct change in the receptor and therefore doesn't lead to the events inside of the cell that adenosine ordinarily is modifying and controlling. And so here you can see, and at first glance it's like, well, those don't look anything alike, but notice that on the left here we have this sort of central ring structure, which is very similar to the ring structure here on adenosine. So this is the part that apparently will fit into the active site of that receptor protein, but because caffeine lacks this other portion here, it doesn't have the conformational change on the receptor, and therefore doesn't lead to the cascade events that adenosine ordinarily causes. That is the end of chapter 5. It's a big one, it's a heavy one, it's one you might need to go back and rewatch things, but certainly ask me questions if you have them, and thanks.